and the audience in the online who are joined from various campuses, teaching campus and also research stations, and our senior soil scientist and the former Dean Madurai, Dr. K.K. Krishnamurthy, who joined online, and also many renowned soil scientists who are waiting for the lecture of Dr. Vadivelu. So really it is a proud moment that Dr. Vadivelu sir here accepted our invitation and who is going to deliver the endowment lecture in Tamil Nadu Agriculture University. Since Tamil Nadu Agriculture University organizing series of endowment lectures, our Vice Chancellor, Dr. Geeta Lakshmi, due to his busy schedule, she could not be able to join today. But Madam is always insisting that the senior professors in their respective field, having the domain knowledge in a field of interest, may be brought into the TNAU for delivering the endowment lecture. So because of that, the young generation could be benefited because the knowledge which they gained in more than 4 decades in the soil mapping, which definitely because the speaker will traveled in different time frame, 3, 4 different generation he passed, and he started there because the soil mapping has been one of the key area which every soil scientist interested to work, because the technology is totally changing every decade. Because when the people started with the uh, simple digestion method, open digestion with acid, now micro digestion and IR digester, many digestion methods are changing, analytical methods are changing from every generation because technological advancement. And starting from simple flame photometer to AAS to ICP, ICP-MS, so much of nutrient analysis also, the different decade traveled in different way, even though we analyze the micronutrients in the plants or animals, plants or the soil samples. So the every generation, the technology is coming very fastly changing, and also the scientists also adapting to different technology. But the person who traveled in different decade and he have the well experience on the way in which soil mapping is going. Now the remote sensing, GIS, so many data came. Even then the conventional method of soil profiling is still a big role in that. So in that way, we are fortunate that we have the senior professors, we have the traveled in different areas, starting from national system to the different Karnataka state remote sensing application center. He moved different places, and his experience definitely will have an idea on that how we can look forward and where we travel, where we are going. So these things definitely will help us to learn. So this young generation of soil scientists, the masters and doctoral students, learn from his experience and make your newer research area how to move forward in these new directions. And also the senior professors are here, came over here, and really it is it is a blessing occasion that this lecture will have a very good fruitful discussion. And also I request all my masters and PhD students, please try to interact, because every time when you deliver the lecture, the interaction is needed, because otherwise it became a monologue. So we need a dialogue session. Because so much experienced persons here is going to talk to you, but you should definitely give your reflections, whether you accept the concept or you want to contradict on that. It should be debated, and it should be then only we have the scientific discussion. So every speaker will like to have a discussion with the audience. So I expect that, because the soil science students are so bright students, so definitely they will interact at the end of the presentation. So that is what we expect. And also I once again I thank all the persons who are in the hall to watch the lecture, and also the people online. So for information of the house, we already more than 3,000 people. We send the lecture schedules, the events well before, more than 1 month before. So everybody waiting for your lecture. And we've been all social media been spread the message, and also we've been recorded in online and also the YouTube live. And this lecture also will be available in future those who are not able to attend today's lecture because of their busy schedule, they can they can watch the lecture after this also. So with this, I once again welcome you, sir, for this particular endowment lecture on behalf of Tamil Nadu Agriculture University, on behalf of Vice Chancellor. Welcome you, sir.
We would like to invite Dr. P. Balasubramaniam sir, Director NRM, TNA Coimbatore to to introduce the speaker.
The most respected today's chief guest, former head of the Institute of National Bureau of Soil Survey and Land Use Planning, Dr. Vadivelu sir, and Dean SPGS, head of the department, and senior professors who have come to hear the lecture of Dr. Vadivelu sir, Dr. Agassin sir, Dr. Akipan sir, Dr. Muthuvel sir, and all faculty members and my dear students, and our senior professor and former Dean AC and RA Madurai, Dr. KK Krishnamurthy sir, who is on online, and other dignitaries in the online and other scientists in the online. Good afternoon to everyone. Just I want to introduce the speaker. That is a speaker is a well-known person. That is he has put a lot of experience and service in the area of soil resource inventory. He is a consultant of the Karnataka State Remote Sensing Application Center and a former principal scientist and head, National Bureau of Soil Survey and Land Use Planning, Bengaluru. He has obtained PhD in Soil Science and Agricultural Chemistry from Tamil Nadu Agriculture University, Coimbatore. He is an eminent soil scientist, served in National Bureau of Soil Survey and Land Use Planning, ICAR, for more than 30 years. Besides soil mapping, his profession included research and teaching in the areas of soil survey and mapping, soil classification, land evaluation, and land use planning. He served as a task force expert in developing Karnataka geological information system tools, governance, sustainable development, and citizen empowerment of Karnataka state. Especially expert in GIS data and its application for various fields. As well, he is highly skilled in Karnataka GIS decision support system application and capacity building program of Karnataka state. He submitted the planning and monitoring and development report to the government of Karnataka. He has published research articles in the referred journals as an author and co-author of more than 100 research publications. With these few words, just I complete my task of the introduction of the speaker. Thank you very much.
Coming to soil mapping, not many soil scientists prefer to be a soil mapper for obvious reasons. All of you know that. But still, anyway, the soil mapping, understanding soil survey is very, very, very essential, because when we work on soils, either nutrition or microbes or anything, we should know on what kind of soil we are working on. With that view, it's very essential for every soil scientist to know about soil genesis, soil characters, and all. So the topic is soil mapping. You know, soil mapping about if you start soil genesis, soil formation, and all. So this equation you can't avoid. This is NE 1941. So it says, because the we all accept soil information is vital, isn't it? Either for agriculture or anything, even for engineering, everything. Soil information is. So now, India, we have diversified geological formations, climatic conditions, geographic features, and vegetation. If some of you have widely traveled, or within the state also, when you travel, keep your concentration on what you see through the window. You see the soil, you see the landscape, you see things. Then you will know how rich we are. So the soil formation says, any formula says, soil is a function of, all of you know this, all scientists know, climate, organism, relief, and parent material. Remember this. Remember throughout this lecture and afterwards. Soil is a function of this. So when we say soil is a function of this, naturally it varies everywhere. Because everywhere the climate, organism, relief is going to be different. Parent material is going to be different. So that means soil varies, every inch soil varies. The different already altogether different matter we group them, but soil varies. Here in this formula, there is another implication. The implication is a soil scientist. Soil, the function of this. So that means the implication is soil scientists should have a knowledge of all this climate, organism, environmental time. If you are working in a state or you working in a country, better suppose we take India as a country, then you should have a thorough knowledge about all this. Yes. Then only we will appreciate the soil what you see, the soil where you are working. This is because I said you should have a clear understanding. So this one, maybe many of you are familiar. Maybe sometimes if you are not, just look at this geological map of India. So here you will find the recent to the oldest it is arranged. The yellow is Pleistocene is the era, the period we can say that is 2 million years. This 2 million years we call the Pleistocene, and you see, suppose you know about glaciation. The globe, the major part, particularly the part near the northern pole, was subjected to glaciation. Glaciation means the ice movement. Ice was totally moving over. The last glaciation was 12,000 years before the last, that is the last one. That means it never reached India, but the north of, so it was the Europe. And therefore they will say it's a glacial till. They'll say the parent material is glacial till because most part of northern Asian continent and Europe was affected by glaciation. You cannot even imagine, like suppose the earthquake. Few days back, there was an earthquake in Turkey. Still people are working on that. Why it happens in Turkey? It's all geology. Why it happened in Turkey is, see, these are the tectonic plate movement. See, the entire globe has some tectonic plates. So these plates, when they rub against each other, particularly in Turkey, the Arabian plate, Indian plate, and the Eurasian. Europe and Eurasian plate, they say, when they come closer and then the Indian plate is moving north, still it is moving north, though we don't feel it is moving north. Every year, maybe 0.5 centimeter or something like that, they say. In this movement, there the tectonic plates move slowly only, but the impact is what we is saying in, that is the impact. Anyway, leaving that apart, so the geological map, if you see the Pleistocene, then we have the sediment. Tertiary period. After the Pleistocene, some people will say Pleistocene, some will say Quaternary. Recent, then before that, Tertiary. So tertiary period from 2 million years to was up to 55 million years roughly, because people may vary some few million years this way, that way, but 55 million years, that is a tertiary period. So during this period, huge changes were happening. But you see the rock formations, sedimentary and tertiary, the northeastern hills. Northeastern hills, all they say the Naga Hills. There is a Naga Hills. I had an opportunity, Naga Hills, nice area. So these are all tertiary period. And the Himalayas and Deccan Trap. Maharashtra, you see the green. Maharashtra, this is what they call is a Deccan Trap. Why I'm telling this is soil scientists should know that. The Deccan Trap is the basaltic lava flow. That is the last lava flow in the part we had. It's a lava flow by the basalt because it happened between the Tertiary and Cretaceous period. Around 60 million years ago, there was a sequence of basaltic lava flow. So that is solidified. That is the basaltic area we say in the Maharashtra, Madhya Pradesh, and northern Karnataka. So this part we have that basaltic lava flow is in the there is the igneous and Deccan trap. Then the some metamorphic. These are all Vindhyan and Satpura ranges. These are metamorphic formations. Satpura range, just to see it is in Malwa Plateau, north of Maharashtra, Nagpur and Tapti. We have Tapti and Narmada. Between that we have ranges. We have that there is one place like Multai or Multapi also. That is where we have a Talakaveri. You have seen Talakaveri, where Kaveri starts, Karnataka. So like that Multapi or Multai, the starting originating place of Tapi or Tapti river is in Madhya Pradesh. So that is the area is see the dark brown. That's the oldest formations. It is a Precambrian. Cambrian means more than 1,500 million to 4,500 million ago when the Earth was formed. So that Precambrian is the second thing. Of course, it stops with an but also same kind of metamorphism. Very old Precambrian. And this part is fairly stable. Fairly stable because we don't have the tectonic plate very close. The Andaman, Northeast, and Himalaya. These are all weak zones because the Australasian, Eurasian, Australian, and Eurasian plates they have the movement. So there, see in Andaman, Northeast, and then Himalaya, frequent earthquakes. Something about the geology of the next, please. So these are all looks like a few million years ago, all that a kind of Walt Disney stories, but still we should know. Then only we can appreciate this oil formation. See something details about it by a Geological Survey of India. Same thing, but more detailed. Next. This for somebody is interested for this period. I said this Tertiary and Cretaceous, that border is 16 years. There only the lava flow of basaltic ended. It's ended during Tertiary Cretaceous. And you see the Precambrians. Cambrians means Cambrian and then for that. Our landmass, Tamil Nadu, the Precambrian means first land. So there is no doubt when we claim our civilization is also oldest. Naturally, in oldest, after that there is no geological upheaval. Stable. So we have our civilization is bit of that next. Then we have the geographic very clear here. And then we have the Western Ghats. See the Deccan Plateau. The Western Ghats are faulted, eroded faces of the Deccan Plateau. Faulted means in geological events, the faulting of plates then and eroded. Eroded and faulted islands. Gangetic Plain is the very recent one. They have Himalayas. Himalayas, the recent formation compared to, so it started with the Himalayan rise in 10 million years ago. Also because of tectonic movement. It was happening through millions of years. But the Himalaya we see here, it's not formed in 1 day or 1 month. Millions of years. Next, very interesting. Yeah. Climatic zones. We have the tropical wet, tropical where we are is tropical climatic concentrate. I had an opportunity to arid regions also. Sorry. So these are the, it may look like why all these things are required, but what I expect is the soil scientists, not others, soil scientists of a university, be very familiar with this. Not though we are working in Tamil Nadu, the Indian geology, Indian geography, Indian physiography, Indian climate is very clear. That is why these are the, we have all kind of desert to kind of Himalayan. And Himalaya also there is a desert. Leh is a cold desert. Is very less, but it is a cold. It's a cold desert we call. And we have the arid desert in Rajasthan and Gujarat. So our part is wet, humid, and dry. So next. Now you see some lines are there with the arrow like these are the monsoons. Monsoon winds. We have the from southwestern part of Indian Ocean. When it comes very close to India, it branches out. One goes to one is the Arabian Sea branch, another is known as the Bay of Bengal branch. The Bay of Bengal branch, it just see you can see that it enters India near Burma. Then here Burma and India is separated by Arakan Hills. It is known as Arakan Hills. Some people say Arakan Yoma. So these are the very big hill ranges. When this Bay of Bengal winds branch, when it hits the Arakan Yoma or Arakan Hills, it is deflected towards the north. When it goes to north, then the northeast and then Himalaya. That's the heavy rains. Northeast, you say it's the heavy rains. Then when it hits Himalayas, then it is turned back. Then the area of Uttar Pradesh, Bihar, the Gangetic region, Gangetic, so they get a heavy rainfall. The Arabian Sea branch of monsoon winds, when it touches the Western Ghats, so most of them checked. Then that is why you see the west flowing rivers in Karnataka and Kerala, they will be full of spatin. Heavy water, large amount of water goes to the sea. So that we can't avoid. Still, some the government is trying to have divert the water back to towards the now this kind of monsoon, they have the heavy rainfall in the western part of Western Ghat, but still the wind with the water, when it flows over, then we have little. That is why Tamil Nadu will have very less rainfall during the southwest monsoon. Then we get through the northeast, and that wind flows from the China part. There we get some kind of rainfall in and during the ocean disturbances. Next, this is something about the monsoon winds and then climate. Next. Same thing is given the pathway of monsoon wind. So you see this last one, when it goes to northeast, hill ranges in Arakan Hills, so it is Himalayas. Himalaya then comes back. Hills of Himalaya changes Brahmaputra. You see, I had an opportunity to be working there in Brahmaputra region. Brahmaputra, we also have an largest river island in the world, Majuli. So, and Brahmaputra is a kind of river. Even people there, they say, when Jawaharlal Nehru saw it first time, oh, not a river, it is an ocean. That was his comment. Because here you see in one bank, you can see the other bank. But in Brahmaputra, many places, if you see in one bank, you can't see the other bank. It is so huge and so wide. The biggest river in India. We can say, they say next to Amazon, this is the anyway. This is our richness, our also same thing about monsoon. Different dates given those and we get per region is split into of rain. Natural vegetation. So naturally, the natural vegetation depends on the mostly about the rainfall. Whenever wherever we get more rains, wherever the soils are good, we have the natural vegetation richness. You see the Western Ghats, and then wherever water is high, you see forest, green, tropical evergreen. We know deciduous and evergreen not going to that. We have both. We are fortunate to have both. The richness, it is just to show these are our prerequisite for a soil studies. The savanna forest, this is a kind of, you just see, we have in our hot and season savanna. So now, that is a background, that's a prerequisite. That's why we say, soil surveyor, before he goes to the field, he should have all this background information of at least the area where he is going. All that, what is the geology of that area, what geography, what are the landforms, what are the rivers and streams, what are the vegetation type, all that we now with all that, when we say soil, it's a very classical definition. All of you know that. Being a student of soil, you know this is the natural body of solids. Then we have minerals and organic matter in that, occupies on land surface, natural, then occupies the space. Space is Euclidean space. Euclidean space is two-dimensional and a three-dimensional unit. So it occupies. There is a fourth dimension, but still we have not come to that people over fourth dimension. But for us, that will be too big. So we restrict to the two dimensions. Then this soil is characterized by, with all that, during the soil development, several important horizons develop. The horizonation. So you dig a soil profile, you see the horizonations. Horizonations because soil forming processes are different. There are something accumulating, something is transforming, transformation. So many things happening, we will see that only help us in naturally it support. So this is kind of a definition. Suppose somebody wants soil look like, you can define anyway, but the USDA, United States Department of Agriculture, follow for many things, including soil classification. Definition is these are the characters of soil. This is how we define. Next. Then this kind of soils we have to map. Why we have to map? We should know the soils. We study and map the soil. We know the soils. We map the soils, and we leave it for others to understand. That is the purpose. So this has been, and the Tamil literature, if you see, soils and the land have been separated like we say, na? Kunji, Mullai, Marudam, Palai, Neidal, something like that. So all those things are soil and land classification. But unfortunately, those things have not gone out of Tamil Nadu. We say we have a very rich culture, but purely we have a very rich. And this kind of land classification was there. But unfortunately, it has not gone out. It is just with us only. With us also means only people who are interested in Tamil language alone will know that, not even others. So because we have not taken this to different level. But what is available in record for us is from 1893. So this is what I say is on Welker and Leather in 98 and 93, they made a kind of earlier stages. They said Indo-Gangetic alluvium, black cotton soils. What they say black cotton soils is, I told, lava flow of basaltic lava flow, that part, Maharashtra, mostly Maharashtra. So if you see, the all soils are black soils, and the clay mineral is smectite. So that will swell and shrink with water. That is black cotton soil. Those days they said it is black cotton soil because cotton was the major crop in that. So it's a black cotton soil. Then red soils and lateritic soils. These are, suppose we come down in the Gangetic alluvium, you will not see this. Then trap, called Deccan trap, Maharashtra. But when you come down, because we say Karnataka, this Deccan plateau and then the east western Ghats very oldest, where the landmass is oldest, red and lateritic soil, because they are stable and so much rainfall and other temperature and the vegetation associated with that, they have been responsible for the formation of lateritic soils. Then after that, several people followed that. Then everybody got interest, and there were people who were interested in the soil. So Vadia is a geologist. So he also mapped the soils, and then the peninsular India, red soils, black soils, lateritic and laterite soils. Then Indo-Gangetic alluvium, you have delta, desert, and Tarai alkaline soils. So this kind of definition, classification and mapping Vadia. Then Vishwanath Ukil, then Rai Chaudhary. He's almost when I joined, he was working. So it's a kind of recent. And then he also did. Then finally, a kind of integrated effort was done by National Bureau of Science because earlier there was no institute like that. Then an institute was formed, National Bureau of Soil Survey. Then the mandate was given to them to map the soils of India. Then the institute took up the job of mapping soils. So to start with in 85, before that some soil mapping was going on. What we did first is to compile those information, then correlate this with a physiographic region, physiographic unit. Then we had the physiographically divided India. Then this was like extrapolated or correlated. Then that way, it is 1985 map was not purely on field work. It was available field work was taken, it was correlated with the landform units, extrapolated to cover the country. That is why here we could not make kind of so much detail. Suppose that is why we made a suborder association. You know, order and suborder. If you know, maybe many of you know that soil taxonomy, your courses. So order and suborder is the next level. We may that time because it is most of the things are extrapolated. We cannot very clearly tell this is this. So at a higher level, the generalized level, soil map was suborder. Later, this is that map, suborder association map, 85. And it is given here, in the is this map and then go to that what are the legend is not very next. Then in 1986, Dr. J.L. Sehgal joined the director of National Soil Survey. He took the very important step of mapping the soils of all the states in India at a 1 is to 250,000 scale. Now, At that time, even to map the soils against 250,000 scale is a huge task. All the states means, you can just imagine. So we mobilized the state departments, took their help, and then the methodology was defined. How do we do this? So we that time, the only satellite image available to us was Landsat. So Landsat, that is also 60-meter spatial resolution. You can just see. Now that is 60-meter spatial resolution. Now we have 50-centimeter spatial resolution. Even higher we will get after maybe. That is the kind of there, not much detail, but even that was the only thing available. That Landsat satellite images. Then went for the physiographic delineation of the entire country. Then here also we can't go for survey everywhere. Here also on defined, on well-defined physiographic units, we studied the soils in detail. Then it is extrapolated to other units. Thus the scale was 100, 200,000. And then right there, we debated several meetings. It was debated. Then it was decided there. You may remember, you may recall some of your in classes telling soil series. So they said soil series level is very difficult. So we made soil family association because the scale is have very direct relationship with your taxonomic unit, what or soil mapping. So it was the meeting. It was decided we will make a soil family association. And the work was done. It work almost went for 10 years. Then huge survey, then coming the cartography work. Those time we never had GIS software and all. So computer was just coming that time. So it was all done through the conventional methods. It was again printed in a printing press. So this map is there, Tamil Nadu map, the department you can. So that was a very major step taken by National Bureau of Soil Survey. It was complete. This is one of the example of the soil map of Kolar district. So we have the next. These are the legion. You see, this is the soil family association. Fine kylenetic typic. Maybe now don't bother about what is Candice Starfish, because now that will be too much now lecture. So two families, fine kylenetic, both are alphisol. They have the mineral and the the exchangeable cations, cation exchange capacity. That different. That is why these two have in association. The two are family. Next, so everywhere the Kolar district, this slide is shown. What press upon you is at that scale, only soil family association alone is possible. We can't go further down. Next, then when we why we should make all this soil mapping? What do we get from that? We have from the soil map and then data we have description and characteristics of the soils, soil taxonomical classification, soil units we delineate and a map, soil properties we get our records. Then with all that we can go for prediction of suitability and limitations. When we know the soils, we know the limitations. For example, a soil may be good for one and may not be good for the other. Why? For one crop, it may be very good. For other crop, there is a limitation. So we can predict this is good for the soil, particular crop, and it is not, it has the limitations, it is not good for other crops. Soil mapping gives us this kind of information so that we can go for our land use. Next. So now, while mapping the soils, how the soils behave or how do we see the soils? Because soils just, it's not random everywhere. Suppose soils doesn't occur in random. Soils, they have a pattern. Why they have a pattern? Because we said soil is a function of so many factors. No? These factors, suppose climate, suddenly it's not that 1 year moist climate, another year like desert-like climate, we don't get. Climate, we see the climatic regions. No? So humid region remains a humid region with a little fluctuation rainfall. Arid region remains arid with the fluctuation. Same arid subhumid regions, they remain subhumid with little. So they don't change. Much changes, it will not. 1 year humid, another year arid, it doesn't happen. When that things changes like that is not happening, the soils also form as a pattern. So we have a regional pattern, large area, regional pattern. I said Maharashtra, the basaltic region, the climate, everything is constant. So we have totally all black soil. That's why they said cotton black soils, black cotton soil. Then Indo-Gangetic alluvium, they say Indo-Gangetic alluvial soils. Nothing more can, because they are the parent material. It's all Himalayas and then Himalaya, that alluvial, whatever alluvium brought by the Indo-Gangetic, it is all deposited. So it's all Indo-Gangetic. Then when we come back here, southern India, red and lateritic soils. So they have a pattern regionally. Then that same pattern continues to local level also. Local area also, the topography, then parent material, time of formation, and we can say the microclimate. So with all that, soil have a pattern of occurrence. So this pattern helps us in soil mapping. Everywhere it is randomly occurring, we possible to, because it soil occur as a pattern, possible to map the soil. Next, please. So as we said, even I said when we made a 6.5 million map also, soil physiographic, soil landscape relationship and then extra. So soil landscape relationship very essential in soil mapping. Then landforms, we say the microclimate, vegetation uniform, relief almost, and relief is every landform, the relief change. Parent material remains almost uniform. Basalt means basalt, alluvium, alluvium. So, and the parent material, in situ parent material, soil. So we get this soil, the characteristic of soil varies with the landform. You can say when we go for local, smaller area, can say soil varies with the landform changes. Just to, this is what I said, when you are traveling, you will see this kind of. Now, that's what I read an article, there it is written, when two persons travel somewhere, or bus, whatever it is a soil surveyor, soil mapper, another. So they both look through the windows what is outside. Then he says the way a soil mapper looks out and then he perceives is totally different from the other person. Other person may say it is a dry or wet or somehow people are there, what is going. But a soil mapper will immediately know what is the kind of landscape, what are the vegetation going there, then what kind of soils we will get across. So this kind of thinking will go on in a soil mapper. That is true also. So now whenever you go, particularly soil sensitive, you keep, don't look into your cell phones. You look into through window and then see what kind of landform and other things are getting outside. And you can even make a note of it every, suppose one day you go from to Kanyakumari and then some Madras, Chennai, then you can see, make a note also. Next, these are the different kind of landform. We insist to indicate just how landform varies. Very local level. This is our eroded surface. So now you see that depending on landform, the slope and other thing, slope, particularly relief characters, are slope characteristics, the soil property. So this is one, for example, you see the soil depth. In summit, where summit means the top of a hill like. Where it is almost flat, you will see soil depth is more. This is very common you have seen. In a slope, the soil depth will be less because it's all eroded. So when you come back to the valleys, you will have a very deep soils. That means we are, here there are so many parameters to consider. For example, I am just giving for brevity of time. I am just telling only the soil thickness or the depth of the soil. You see, with soils vary, different relief or slope. Emphasizing so landform studies, landform is very essential for soil mapping. Next. This is one three-dimensional. You can see it is taken from USDA manual. I don't know, you have the habit of reading. USDA has a soil survey manual. I don't know if that is available in the department. So that gives, it's not only about soil mapping. You just read this, you will understand how what soil is. The manual prepared by USDA, both soil survey manual and then soil taxonomy. And now they say keys to soil taxonomy, yes, abridged version of soil taxonomy. These things every soil science student should read. There it is taken from that. You see the different soil series, you see the locations, kind of similar landforms, they are getting repeated. One is the valley, both sides ridges, and then the slopes, and then the valley, soils are same. Like, you see again to emphasize, soils mostly in the local level, soil formation, soil character depend on the relief characteristics and form. How do we proceed with soil mapping? You know, so earlier we used to do, now also some people conventional soil mapping, kind of assessment, personal assessment, experience. They will take soil profile. Then we have also have the physiographic delineation using topographic map. Then we never had anything else. Only the tool available with us was Survey of India topographic or topom. So with that, we move around in the field and then take profiles and then get to the help of topo sheets to place the boundaries. Now soil mapping, what is important is you will study a profile. All that will be very fine. Excellently, you will do. Excellently, the soil samples will be analyzed in the lab. You will have some kind of 100 profiles you have taken. The soils are varying. Now, where do you place the boundary? One soil here is different, another soil is different. So, 100 meter that side or 1 kilometer that soil different, this soil different. But where do you place the line? Where do you separate? That is a part of mapping, isn't it? That line, where do you put? That is very difficult. Earlier, we were using seeds to drainage lines to place that line. Now, edges, so there, tonal difference. See, after all, a tone is the reflective characters. So the electromagnetic spectrum from the sun getting something is observed, it gets reflected. That reflected spectrum is sensed by the sensors which stand in the cell. So that then that is converted to our images. So they have the different colors, tone. We can use the tonal difference to raise the bound. Then we can go for. That's what we do, study the soils and then classify. Naturally, so when we do this, for kind of mapping, soil surveys classify polypidon. And we soil mapping. Soil polypidon in the unit or 3 can use. We get, you have seen, I think most of you have seen satellite images, remote sensing GS classes. You have seen or no? See, most of you have seen how a satellite look like. So this is how it look like. If we using this, we go for tonal differences are there. You can see. So we make units, and It is possible within a delineation, 1 or 2 or 3 soils are occurring. Next, this is a one such delineation. In Karnataka, we made one area. These are the satellite image. We made these lines, green lines. You see, these are the based on tonal difference. Tonal differences, the units were made. You can say we have delineated a soil body. We don't know what a soil body is. We delineated soil bodies. Why we delineated using the tonal difference? The one soil body is a different soil body. At this stage, we know we differentiated the soil body. What is that natural soil body? We will do. So the what is important is using the satellite image, we delineate the natural soil bodies. After delineating that, we go for the profiles. Doing that, when we study, we identify the soils and then classify it. So identifying and the classification that we will now in this lecture we will not cover that. Need array sometime not like so formal. Some informal classes I don't mind coming and speaking to students, but here we will not go into the details of soil classification. And that for obvious reasons that is too huge to cover now. So we delineate the individual soils, then the pedans. So pedan definition may be all of you are aware of it. Pedan is the smallest soil body. So there is one figure to illustrate that. Go to next. Now here you see we see this column, this individual pedans. Then you see this box-like thing. It is written there polypedan and another polypedan. So this kind of small pedans, many put together because one pedan they say it's by definition it is not more than 10 square meters. So we cannot make a soil unit for 10 square meter. 1 square meter to 10 square meter we cannot map the soils. So the similar pedans are placed as a polypedan. In the definition, poly, it's many. So many pedans which are similar. Or why they are similar? Who says that is similar? I will not tell we are similar. No? Pedan will not say I am similar to other pedan. How it is similar? We define ranges. When we make so how we define ranges for use. Our use is mostly agriculture and crops. So we define the ranges. For example, cropping in keeping that in mind, we say, well, okay, some soil is 20 centimeter, another soil is 28 centimeter. Pedan. So we are not going to differentiate that. Another is 15 centimeter, we are not going to differentiate that. We will have, okay, up to 30 centimeter, I will put in one group. That we decide. So after deciding that, we say, these are so, though they are several pedans, they differ, all that we group as a polypedan. Then again, here you see one polypedan, another polypedan, there is a red line separating them. There also how, again, we fix limits. We fix limits, okay, we say up to 30, we accommodated in one polypedan. For our purpose, maybe we thought, okay, above 30 centimeter, maybe up or 50 centimeter, I can accommodate in another class. So we can say this one red line this side, we fixed that limit 30 centimeter depth. It is just an example I'm giving 30 centimeter depth. Similarly, other properties. So this way we study a pedan, then we group polypedans because polypedan is easier to map because we accommodate some ranges. We get in considerable area, so we can map that. So we make different polypedans and then therefore the polypedan is a mapping unit, like we can use it to map. And this polypedan in other ways it is called soil series. Let's say soil series, when we say Coimbatore soil series or Palladam soil series, the soil properties we will write, then I don't know, you are all exposed to that or not, but soil series, we define the range in character. It is not that Palladam soil series 35 centimeter, it is only 35 centimeter depth. No, it will vary from 30 centimeter to 50 centimeter, 60 centimeter. We give fix that range. Within that it varies. Clay content varies between this range. Silt varies between this range. Cations vary between this range. This kind of ranges we fix, and then we are when we write a definition of a Coimbatore soil series, after typifying pedan, we say one pedan character after describing. They also write the range in character. Without range in character, soil series description cannot be complete because now you see polypedan, there are several pedans. We can't give one depth or one CEC or one clay percentage. So we have to accommodate a range in that. So that range is very essential. That range we fix it depending upon the what use we were putting the soil in. Something about the definition about polypedan is used to define a soil series, a unit of soil mapping, because it will cover a considerable area. So we can use it as a unit of and homogeneous at series level. Why? How homogeneous? It's not homogeneous actually. Homogeneous with reference to the limit we have fixed. Nothing like the soil is not will never tell what I am and how I am differing. The three fixed limits, and in that limit it is. So we have collected descriptions and all. Then when we say we will slightly move to different now up to the now is the soils how we have pedan and polypedan we say. Then how do you actually we map it? This is one soil map, Karnataka. Here you see these are the different soils and then soil units are given. So this also these delineations originated from the satellite image. Satellite image and to make these definitions. Then the soils are studied here. We go there and study the soils. How is it one soil? Because this is a very large scale map. Large scale map means we said 1 is to 250,000. This is 1 is to 8,000 only. So why 1 is to 8,000 is large scale, 1 is to 250,000 small scale? It is large area, the ratio. Ratio 1 divided by 250,000, 1 divided by 8,000, which is big and which is small. So that is using that we say small scale and map. This is a very large scale map, only on scale it is mapped. The delineation means the line, the boundary line. The delineations are made from satellite image. Next. Then the soil map units. We have different kinds of soil map units depending on the purpose of survey. Soil map units always it depends on the purpose which we are studying, the pattern of soils and then miscellaneous area. Depend on all these the purposes. Next. So these are the map unit component, the essential. I said we delineate the natural soil body. So within that soil body, what are the soils occurring? These are the components. Either you seeing the satellite tonal differences, you had a delineation, you boundary. That is a natural soil body. So when you see inside there can be miscellaneous area also. Component within that unit created can be a soil or soil also. One can be a major soil, another can be a minor. And another can be some miscellaneous area also. So these are the map unit components. Map unit, a soil, a miscellaneous area, major unit of soil, and all these things. We call that a component. So that's why the component, it can be a soil series or the category above soil series. What are the categories above soil series? You remember? Soil series is the lowest category in US soil taxonomy. Category above the soil family, then above this subgroup, grade group, suborder, and order is the highest category. So that map unit component can be a soil series or any of the higher category. Then some tax adjuncts, this we rarely use. This kind of soil looks like similar to another kind of soil we have already named, but beyond the taxonomic class limits. So when you work on that, you, otherwise this will take a little different. For example, we have to study a INTP Caplostat soil series. So another soil looks like almost like this, so we use the name. But why they are tax adjuncts? Because at higher level of it is going outside the range of classification. So tax adjuncts, theoretical interest, we rarely come across this. Then miscellaneous area, then the SS of component. SS means a same soil series. Sometimes suddenly for some area it will be some undulating. Some will be, it's a palladum soil series. Okay, it is a plain land only. But it is possible some places for some reasons it is undulating. There we say it's an undulating palladum soil series. So all these things are possible as a map. Being very big, there is a delineation made. Within that, the components of the within the delineation can be. So we say soil series because mostly soil series is used. That is polypedon. You know, by time soil series is polypedon. So we differentiate when we field work study. We start studying soil profile, soil series. Morning there was kind of field correlation done first by the soil mapper himself. I study a profile here. I say a particular soil series A. When I go to next place and then when I study, it is close to or similar in that range. I give the same name or I keep giving different name. So with the differentiate soil series based on this. If you had some time gone in the field and then studied profile, you will know these are the different properties. Based on that, we make differences, differentiate between one soil to other soil. What are these? This will be too much again here. I cannot start explaining what are the different structures, redoxymorphic features, all that you know already from. If any doubts, your teachers are here, and so you can approach them to all these things. So anyhow, based on these soil characters, we name the soil series. Soil series. Then when after identifying soil series in a map, identifying, we made a map. Then there are map units. There are kinds of map unit. One is a consociation. Mostly we use associations because the soils they occur in association. We say one polypedon, another polypedon. In we said there is a delineation natural soil body we delineate. Within that there are two soil series, two polypedon. One is major, another is minor. So we place both of them. So it is an association. Mostly in association, other things are very rarely we come across complexes, and association is the very common kind of the map unit. Even when I say so the Kolar district, you see they are in association. Two soil families in because we cannot separate it out. We have to put it within that delineation. Separating that is going very difficult at that scale. If you again go for still larger mapping, you can do it. But the scale we preferred, it will not be possible. Make it together as an associate. Next. The same three-dimensional figure I am repeating because these are the soil series. Some 2, 3. Then for this kind of soil mapping, what we require is these are the information. We need site information, farm drainage, everything. So we also give you a form wherein we have to write site information. So these are the site information. What is the parent material? What is the bedrock? That's why I said when you are required to write all this. To write all this, you should be knowing about this. If you don't know what is the biological parent material there, you cannot write. So all that is required for a soil mapper. Then soil information in the field, we study, these are the profile study. Normal profile study, we give of depth, color, the horizons, diagnostic horizons, all that we speak about, we make a note of it. Then sample is taken to the field, then laboratory, sample is taken to the laboratory, and we go for estimation, a set of estimation. So field information, morphological, what we call is a morphological study. When you are in the field, the morphology, what you see, you are writing. More than that. Then you take samples and then bring those laboratory studies, both the morphological, then physical and the chemical characteristics of the soils you get. Then all this will help you in classifying the soils. Then so these are, sorry, we cannot go details of what are these requirements, what are these, because then it will course. So you may, I think I have a course in soil mapping, I think, soil genesis. So there you can clarify all your doubts, or you can read yourself. Also you can understand, because you are all in a program, read and understand. And anytime want to my intervention, I can even. Next. Then soil taxonomy is another one. The classification. After getting all those soil characteristics, then we place them in some classification. So what we do is we have, in short, the have diagnostic horizons, both in the surface and the subsurface. The term itself, it's a diagnostic. How it is diagnostic? We said factors of soil formation. Then with that, the different process of soil formation, factors and processes act on the soils so much, we get some diagnostic features, we can look at it. We can diagnose some features. Then and with analysis and also morphology. These are diagnostic horizons. Some diagnostic horizons are in the surface that we call epipedon. Some diagnostic horizons are in the subsurface. We say they are subsurface diagnostic. So what are they? No, in detail. For the most common epipedon for our region is mollic, acric, and these are the commonly occurring in our area. These are the surface horizon. And subsurface, we have this agric, argillic, calcic. For example, argillic is very quite a common subsurface horizon. Argillic means where the clay from the surface, it has moved, removed from the surface and then deposited in the subsurface. This is argillic horizon. This is a very similarly gypsic, natric, oxic. These are all natric where the sodium accumulation is there. And for example, you know the alkali soils, the ionic index, when it is saturated with sodium, sodium-rich soils, then that is a natric horizon. What will happen when the clay is charged with sodium? It will not come together. See, structural formation is a very one of important features for crop growing. So structural formation will not happen because one clay mineral will repel the other one. Same is saturated. So there will not be a good structure formation. So sodium-rich soils, no crops grow only for this reason. Sodium actually is not bad to the crop. Problem is this one. You say the flocculation and deflocculation. happen there, and there is a natric horizon. So this and salt accumulation. All these things will happen because of the soil forming factors, soil forming processes. Soil forming processes are governed by. What is important is for classifying soils, the diagnostic horizon. Horizon itself is diagnostic. We name this as the characters of diagnostic horizon classification. Now, anyway, this is about the conventional soil mapping. Soil map. Why soil mapping is required? How do now earlier we never had satellite images. We started on that. Later on, when Dr. Segal came, I said we had a Landsat image, which is 60-meter resolution. Very less details will be available, not higher details. Using that we did earlier. Before that, even that was not available. So soil mapping, particularly the placement of boundaries, was very difficult. We are, see, there individual judgment. I make the boundary line here, centimeter apart. If somebody else making, I cannot say he is wrong or he cannot say I am wrong, because there are left to the. Now, when we had a satellite imagery, things were made easy. All differences we can use, and then we can delineate the soil bodies, then go for identifying the soil. That is one part. That's a conventional soil mapping. Then digital soil mapping has come with all the technology improvement, and I am happy. I think TNAU, the remote sensing and the GS division is strong. That's what they are also mapping. This one is told me we are also digitally mapping soil. But you remember one thing, even if we go for digital soil mapping, we use satellite images. What we do is, there also the actual field study is very essential. What we do in digital soil mapping, we have already the soils studied, field and soil studied. Then with image elements, we are just correlating the information already available, either from soil map or profile characteristics, or you go again and then study the soil and then correlate. We go for any kind of technology, even if we go for technology, help of tech, field work, we cannot repeat. Because to validate this technology, field work is very, very essential. So this is why you can just see this digital soil mapping. Next. Again, you see the same thing. We use soil is a function of climate, organism, relief, parameter. It's any 1940 classic. But the limitation here is, these are all qualitative. Qualitative, they are not quantitative. We cannot quantify the factors given here. Cannot quantify. We cannot differentiate. All those things are possible. But the same thing, same formula, it was revised by the people started digital mapping it. When we say there is another, it's a Chloropt model, we say another one is Scorpion model. So the difference here is, you see, soil class or soil attribute. You can say a soil class, but soil class is a function of soil, function of the soil itself. Then all those organism time and so what is the change here is, the important change here is, early here in the Scorpion model, all these factors are environmental covariates. This is very essential. The covariate means change in one will influence another. They are all together, interaction. Otherwise, we say interaction, A and B, character of A, character of B, and then there is one interaction effect. So that is what it is based in the Scorpion model. They are all not environmental variables, they are environmental covariables. It is considered a covariable. That is why soil again is a function of all this. And there the soil itself is one of the factors. So this improvement, this is essential for soil mapping. So interaction earlier it was not considered. In this map it is considered. So why soil? Soil at a location, for example, XY coordinates. You also go in the field and then you collect soil sample, you the coordinates X, Y, lat long. So these are the coordinates. So soil at a location of coordinates depend on geographic coordinates, the location plus the neighboring soils. Why the neighboring soil is taken? That is why for digital soil mapping. Many elements, neighborhood, there is one concept of neighborhood, fairness neighborhood. So that neighborhood, suppose the soil into one place XY is also dependent on, it is dependent on the soil nearest neighborhood. That there is one terminology like this. The remote sensing people will know. Nearest neighborhood soil. It is very, for example, my health, my welfare depends on the nearest neighborhood. Isn't it? Nearest neighborhood is not okay, I am also not okay. Same thing in soil also. Soil so depends on the nearest soil. So the soil, say soil is a function XY, soil location at XY is dependent on soil at X plus U and Y plus V. Plus U plus V is the neighbor. So now when you revise and rewrite the equation, soil at XY is a function of XY itself plus the soil at X plus U and Y plus V. It means the particular soil itself, then the so this concept is taken. Why we should do this? Because we are correlating. Everywhere we cannot bore the soil, land, and then take observations. Somewhere we have to correlate. So for correlating the nearest neighbor. This is how in digital soil mapping, you sometimes I don't where digital soil mapping is going on. Ask them to ask also classes to for all of you, for students as well as teachers. Because you should know what is happening in the university with particularly when it is concerned with the soil mapping, soil scientists should know. Next, please. So the square fund model has some advantages because it is in a quantitative, so we can go for predictive models. So these are all possible with the digital soil mapping. Yeah, that's the only advantage over that. Yeah, there are several steps. What is important is you can predict, you can go for quantifying. But in all that, in the earlier model also, the NIS model was given to the function of chloropt there. We know for soil these are all essential, but we cannot go the reverse. Of knowing these factors, we cannot fix the soil. But here, scorpion model that is possible. NIS model chloropt, that is there because it's a qualitative. There, from that for particular soil, these are all important. But we cannot correlate if these are like this, the soil can be like this. First way we cannot come. But here that is possible. Predictions we make is digital, that is PowerPoint model. We are slightly shifting to all the digital soil mapping. Also, technology has developed. So it has made things easy. Naturally, slowly, the geographic information system is what we call the global positioning system, GPS, what we call, and then remotely sensed satellite spectral. So then again, using spectral data, satellite images, elevation are available, and again some softwares. All these things are helping us to go for digital soil classification. How much time I have? Go some 15 minutes? Because it's a weekend, I should be careful. Because all of you will have several programs after this. Some may be even going out of all that possibility. will run through. Here, it is only now the purpose of lecture not to explain everything, but just to indicate these are all there. Now you should go for further reading. Only for that. This what I am telling you. Now these things are the modern tools available in the hands of soil mapper. Next. So remote sensing, we have the digital spectral data. So I said using this we go for soil mapping. Next. This is a kind of some classical examples. Now you see the reflectance spectra. See the vegetation. The green line is the vegetation. This is the spectral reflectance curve. You see in the visible region, the green, there is one small peak. So that is the peak of vegetation. Suppose that visible region, there is one tree as well as one plastic tree. If you take a photograph of in the visible region, both will look green only. But you use the spectral data in the near infrared region. Near infrared region, the plastic tree will not give anything. Only the green vegetation will give. Point you know. Because of it's a chlorophyll carrier. So we have the to identify, separate out vegetation. They go for not visible region, the near infrared. And you see the soils also similarly. These are the kind of spectral signatures to help us in identifying the soils, vegetation, these things. So this is the kind of spectral reflectance coming to our help, soil mapper. There was a program to develop a soil sensor. I don't know whether there was a project also developing soil sensor. I don't know what happened to that, but this is some classical example. These are the wavelength and then reflectance spectra of vegetation, bare soil, and vegetation is almost very. It doesn't give any. It's almost like for some up to visible and near infrared, it's almost like soil. There is no chlorophyll. Not to exhibit any character of any reflectance. Next is the educational slide. So how do we get sunlight? So it some absorptions have a surface. So some energy is used. What is coming is emission plus reflection. So that is taken to sensors. Sensor again to gravitation. So what is happening? Remote sensing. We have our NRSA doing this kind of getting the signals, spectral signatures, and making processing them, and then giving satellite images to the country. NRSA is taking. Whenever we purchase through them. Next. Electromagnetics. Where we are concerned with what we see is the small range. Otherwise, the range is very big. Electromagnetics. Next. So the resolution is the they have the pixels. So the pixel size is the 10-meter resolution. Or we said pixel size is the sensor's characteristic. Then when the sensor is highly developed, now we have 50 centimeter. Pixel size is one. We have list 3. I think yes, kind there our NRSA. Are the characters. Are in red and plus one and that is auto infrared. This is one blue, green, red band, then one SATA infrared. The list for image, a little just 5.8 meters. So we get more information from. Can differentiate either whatever vegetation or the whatever surface or objects we see, can differentiate better using this image. Next, 5.8 meters. P2, and this is green, red, and the near infrared. So these three bands, the other Indian satellites, XORSAT, XORSAT 1 and Cartosat also our Indian satellite. They have 0.5 meter image like this. Spatial resolution is 2.5 meter. It's kind of synchromatic. Next, Cartosat 2, it's 1 meter. Not available. Not available readily. I was told if you ask for it is available. Next, there foreign satellites. Purchase foreign satellites also we purchase for purpose because these are 50 centimeter spatial resolution. So whenever we want to map, we see the roads, everything is clearly visible. Uh, so many purposes. We go for very high resolution satellite image like this, but expensive. Next. So these are the what is GIS. I think you also have many people have a course GIS, so it's nothing new. Things are available in the department. They have a print, can give the printout. Some basics, these are all some basics of GIS. What is important is we deal with the geospatial data. Geospatial data means geographically referenced data. Everything will have a coordinate. It is the coordinate system data. Without coordinate system, we cannot say it's a geospatial data or we cannot use it geographic software. Next. So what we do is actually the geographic information system, we use the geographic phenomena. Or, for example, see in simple words, for example, elevation. Elevation is there everywhere. Suppose you go out and then altitude. So for example, if you measure altitude, every place will have an altitude. So that is a geographic field we say in GIS. It's a geographic. It's a continuous. Any point on Earth will have an elevation altitude. So it's a geographic field. There is another one geographic object. Suppose we say agriculture college, golden building is a point. Can say or when we go for in higher detail, it can be a polygon also. For many purposes, in a small scale, we can say the thing is a point. So there we can say it's an object. It's an object here. So in general, in short, we can say the man-made things we say geographic objects, and then the natural things are like elevation, air temperature. For example, the sea, the in salt content, something like that. These are all geographic fields. Next, this is what we do, but these things we study only about this geographic field and geographic objects. But how to represent this in the computer? Fine, we say it's we know it's a elevated elevation is our soil map is there, land use map is there, crop map is. It's all fine, but how do we put it in a computer? How do computer understand this? What do we do? Next. So this is, for example, this is an example of elevation. These are the, you can see everywhere it will have an elevation, but everywhere we cannot make observations and then use it. Next. One is the continuous field and then discrete field. Continuous field, the elevation we saw continuous. We cannot a line here only the elevation stops here at a different elevation. On a discrete field where we can put some boundaries. For example, one crop field is there, sorghum field. Sorghum, we can put a boundary. It's a discrete boundary. Next. Then I said, how do we represent in the, or take it to a computer system? So that's why we take a raster, because any area, you make some small cells. The cells will have a, we, we, we feel, or we say the one cell has one value. It doesn't happen that way, but even then somewhere we have to make some generalization. So a field you make into very small squares and then say this square will have this value. We fix like that because somewhere we have to do some manipulation mapping. So for that we call the raster, or it's called a tessellation. So you make this, but here also we give one value for that. One cell, we say this elevation means it is 250 meter. That way we fix it. So this is one is the representation, like this is a raster format, or we say tessellation. Next. Then another one is vector representation. So when we do, then we make lines. How do we make lines? The vector is coordinate based. For example, one dot anywhere on the earth is x and y coordinate we fix. That is a vector. So when vectorize your map or what, say when we say vector, it has a, so we vectorize it, and then that's why they say a vector has a georeference. So whereas the raster will not have, because that one square we assume is one, but the raster also has an coordinate, that is the left bottom. Left bottom we give georeference, that we fix that georeference belongs to that square. Anyhow, so these things are done somewhere. You have to make some compromise. But the vector lines is all like everywhere we have the XY coordinate. That is also sometimes you digitize, you will make one click here, another click somewhere, you go somewhere. So in between there becomes some kind of approximation. So when you actually work with GIS software, you will understand. So here vector representation, the point, then line, and then area we represent. Because these are the only things, you know. Point we represent, line we represent, area we represent. So the in 2D system, a single point will have x and y coordinates per point. Every point will have x and y coordinates. So if you say Coimbatore also, Coimbatore, you say for any area, we cannot make one xy. They will say from xy from north this to south north, then this east. This much of 4 coordinates we for an area at least. For a point, we give line will have this kind of coordinates, some vertices, internal nodes, and vertices. I don't know, maybe this I introduce because purpose is not that everything explained here. You go for further reading. Represent raster is represent. Please go and along with that, you go for using GIS software. Suppose GIS software not available, you get the help of our remote sensing GIS division. Open source software for QGIS. GIS software is open source. You can computers you can download and then keep working. Next. So this is a polygon representation. Here the advantage is the entire line is not, suppose you take one line, the polygon separating one from the other. It is not that everything, because once you digitize, it will again for the other polygon also you need not. When you do this, then what is left and what is right, the software takes care. So that is how the area is calculated. So it's little detailed discussion will be required for this, but that's what how points are represented, how lines are represented, what are the different, and similarly, again, what are the vertices used in polygon, all that you. We have map and resolution. We already saw the scale of map, and then the resolution is pixel size. Even that you see, even the television when you purchase, we use the pixel size. Somewhere in the earlier years, you will say the resolution was not that good. Now, whatever TV television set from 10,080 or to 1,000 something, that is a pixel resolution. Next. So it's when we go for the GIS, the spatial referencing is very important. Spatial referencing, they like coordinates, X, Y. Coordinates, these are very important. But the Earth is, we say it's a spherical, all that, but Earth is a very awkward picture it is. Somewhere hills, somewhere valleys. So how do we bring this kind of Earth into a map? That is a big task. You remember, just imagine. We see a map and then we say we have a map. Very easily we say we have the map. How do we get to that map? The Earth is kind of, we say a globe and all we see. There are elevations, hills, valleys. How can that be? For example, Himalaya is so high, Gangetic Valley is so low. How can Earth can be not? But we make that for some purposes. Because we have to work with this. So we make, so that we call the reference coordinate system. And then coordinates from one system, one software to other. This area to coordinate system also, it is not common throughout the world. Reference surfaces also not common throughout the world. From one reference surface we have to transform. So these transformations are kind of falling not easy. So again, the satellite-based positioning, GPS, what time we use. These are all kind of little detailed studies. I will see this picture. For example, Earth surface, top line, the Earth surface is like this. See, it will be undulated and downs, all that. There is one geoid in blue line. Just you see that is geoid. Geoid is, we imagine the Earth surface, because it is land, it is undulating so many. Just imagine the Earth surface is totally water. Not going to happen, but if it is covered, how that will look? Totally water covered, still it will have gravitation. That gravitation will be governed by Earth mass, the center of Earth mass. Center of Earth mass, then the water surface, because there will be gravitational. So how that water body around the geoid, geoid is, that is, then geoid also it is like very peculiar this thing. Then we need some mathematical calculations we need, that we will see. For several reasons we need mathematical calculations. So for that we make a kind of mathematical figure, sphere. So with that we work. That is only that mathematical figure only you will see it's a kind of round or ellipse or side, all that. Smooth like, but actually, now next. It's a geoid. It is also exaggerated. So kind of it's not a smooth by a smooth air or anything. It's all like ups and downs. Like you, this is geoid, for example. This is geoid, in fact, means the earth is full of water. It is only pulled up and down only by the center. That this figure will be like. So, and this geoid, why we do is for getting the altitude height of some mountain, height of some building. So height means there is one x-axis, there is z-axis. So that calculation of that z-axis, the height, we use side. Then for India, that is why the mean sea level we say. Mean sea level also, it is not the same level everywhere. Arabian Sea and Bay of Bengal is different. But even then, we have to have some one mean sea level. So that mean sea level is in Chennai. You know that mean sea level for India, it is taken from Chennai. That station is there. From that, the elevation starts for the entire India. So somewhere, some hill in Madhya Pradesh, we say it is 1,500 elevation taken from this sea level. For Z-axis, taken from mean sea level. Chennai is mean 0 there. Then how that is good? It's all survey only. So will not go to those details now. Next. Then we said for Z-axis we use this. Then for, what do we do for X and Y? X and Y, that is not good enough because that geoid also will not obey any mathematical calculations, formula. Then we need some mathematical formula for X and Y coordinates. Horizontal datum, we are horizontal. We need some figure of Earth. So we taken is known as an ellipsoid or spheroid, but an ellipsoid. An ellipsoid. The ellipsoid will have a kind of the horizontally going that is semi-major axis, but dark blue horizontally going that is semi-major. Is major, half is semi-major in the center. Then the vertically going is semi-minor axis. You see this kind of figure, and then there is a kind of flattening is there, isn't it? See the figure, there is a flattening, flattened like that. So semi-major, semi-minor, and that flattening taken for making the coordinates for X and Y. X and Y coordinates. Let's say the the formula. Now you see 6,000, roughly 6,004 semi. For example, that means the diameter is divided by 2. That is the diameter. All these Earth here for mathematical calculation, what is only this much important for y-axis, z-axis, the geoid, which is for x and y coordinates that we use ellipsoid, the reference. Again, this reference ellipsoid also is not globally on everywhere. Every country they have their own ellipsoid. Every country they have their own geoid. That is why if you take a data from United Kingdom and then we use it, then we have to make some transformation. We have to make some transformation. All this now international time that is already. Yes, people 64. Then these are different reference surfaces. Latitude, meridian, and then better for this x, y, and z axis. We will skip this, but you just go to read from some lectures to the reference systems. Terrestrial reference system being used. Why these are all for the GS people, they will use it. But when they use it now, every software has so much tools, you need not to know it. You just, there are tools with every software. You use it, it will do for you. The work is done for you. That is how it is. Now it is made, things are made so simple. But the theory behind that, it's better we know that. Even the coordinate systems, you see, this is like that, calculated like that. Then when you go longitude, then the latitude from 0 to 90, and these are used by the mathematical calculation used ellipsoid. Little this like we should have a class. That class one, for example, this year only we spend 1 hour. Then it will be good. It's easier. For I don't know, for this kind of lecture, this will be too much. But why I am showing all these things here, you should go for that is the only. So this is Cartesian coordinates x, y. See, when we are having for the 3D figures, x, y, we saw the degrees. But here we go for the Cartesian only x, y. So 2D Cartesian coordinates where we go for some meters or whatever it is, like just like graph sheet. Origin, this any geometric lesson or we have seen the x-axis and y-axis. A point will have how much. Suppose we give some values here, how much it will be value in the there. One is the origin. So Cartesian coordinates, 3D as well as 3D is for the globe, 2D is for the and then we said the projections and map projections. So this kind of earth, how do we map? That is it is a kind of a big task. So they project this with this kind of the cylindrical projection. You keep that and you project with a light like. So the lines will come on that. Then you open. For example, we given oranges in your hand. So you take the peel out and then put it flat. How do we put it flat? So it is so tough. So that is how this kind of several projections are made. Every projection what we made is the conical projection. Indian Survey of India uses polyconic. Polyconic Indian government uses big process. This and then traverse cylindrical. This is how the projection is made. And this what use DS universally many people DS softwares use traverse method. Not that everything is fine here. It also has a lot of limitations, but it's better than other projections being used in DS software. All that whatever the terminologies, my purpose is only to introduce these terminologies. Please go for further reading. Or where we get a time, we will not deal so many things at only one thing in a classroom. We can have that. Then the GPS, what we are using, again, this is just to the GPS. There are satellites. United States government, they have satellites. They have 6 orbital plans. In every orbital plan, 4 satellites continuously moving. They are almost kind of more than 20,000 kilometer high. The purpose is now you go out and then you switch on your GPS device in your cell phone. What we have is a GPS receiver only. What we have is a receiver. Those are the GPS satellites. At any time, we can see 4 satellites looking at us or we are looking at it. That is how we get these where we are the location X, Y, and then the Z axis also we get because of the satellite. They are continuously emitting signals. This is also processed by them. Then our it is a time. From that distance, every satellites there sends a signal. Our cell phone also, our receiver has that chip which calculates from that it also sends with time for how much time it has taken to travel our cell phone receiver. So that is calculated. For example, any you in a place you put one dot, and then from 3 different places. From this place it is 5 kilometer, another place it is 8 kilometer, another place 2 kilometer. If you say that, then we can put that point. With 2 it is not possible. With 3 it is possible. 3 satellites signals we can XY coordinates we can find out, but for the Z coordinate we need the 4th one. So they are continuously, these are US military satellites. For military, they have a different band of sending. But this is for the common use. Common public, one band. For military purposes, they have a different that is not available for citizens. So that is how these satellites function. And several countries also they have, but mostly everywhere because this is a well-maintained satellite constellation. So these devices used. How do we use? There is a clock, and this our cell phone chip also has a clock. So to measure how much this time it takes for the electromagnetic spectrum to reach. That speed is known to everybody. Then what is the time it started from? It comes with that information, and what time it reaches our cell phone. So with the time, the distance is measured. So there are different places, some 3 satellites somewhere. So from every satellite, how much time it takes. Then you know what is the position. It all comes with the positional information. They have the positional information. So when we reach our position calculator, this is how this all our cell phone. See, even suppose you Google Map from agriculture college to railway station, they will suddenly there will be red zone. No? So because these people Google everywhere we use the Android phones, so there are some 40-50 cars traffic jam. So all 40-50 cell phones will be sending signal. The Google Map will show the red region. After some time, when they move out, then you will have the. So this is a kind of right listening system that's making life very easy. So this is how function. Please go for further reading. This picture will show how do the signal from the satellites essential to locate one. With 2, it is not possible. With 3, it is from a satellite ABC. In all the 3, we know for satellite position, we know. From that, the signal coming, the time we know. So all that your cell phone has a same kind of chip, same kind of clock, so it will calculate. But even then, this one, what cell phone, the accuracy level is anywhere 15 meter. That difference will be there. So for to still refine calculation for differential GPS, there are some instruments available. We don't need. That's all. These are soil mapping, plus these are the some tools some handy for soil mapping. These are all used, but kindly GPS, satellite remote sensing, JS, these are all a PG course itself. So it's I know I have not justified this because sometimes I am held because where to start, where to end, I was not knowing. So, but still I made this to just to introduce this other thing. Now, as a senior, now I only consider I am a you are senior only. That's all. You are in college. A few years back, I was in the same college. So now what I would request is go for reading, and then I gotten some kind of idea. Your PhD, all that. Some of you, if you interested in technology, go for MTech in there are several Anna University offers, Dehradun offers. Go for geospatial MTech technology, so that your understanding of the much better you can use that, and you can be elsewhere also. You will be useful to the society, remaining taking job elsewhere. So with soil science only, I would not recommend you stop with this. You go for PhD. Some of you interest special, which is kind of picking up because now without these technologies is very difficult. Even I can say I am also learning. It's not that because morning only I told when I was here as a student, nothing was here. So it is a learning process every day. So we have to learn use some of geospatial technology. That's what I would recommend students. Teachers also they should get trained in that. to keep updated. Thank you very much. Again, I thank for the opportunities given to me because, as I said, I never expected Madam Santi will make this so formal, but it's anyway, it's good. He said, no, sir, this is our honor that I am also honored by all of you because you heard me patiently. Thank you very much.
Thank you, sir, for provoking lecture. We request our Professor and Department of Soil Science, ma'am, to deliver a vote of thanks.
Hey, good evening to one and all present here. Respected Director Aram, Dean SPGS, who is busy with some official commitments, and today's guest speaker, one of the senior soil scientists in the country, and our respected teachers, guides, Augustine sir, Rakyapan sir, Dr. Muthuvel sir, and our former Dean AC and RA Madurai, Dr. K.K. Krishnamurthy sir, who has joined online, and many other dignitaries on the through online, and the faculty members of the Department of Soil Science and Agriculture Chemistry, and other faculty members of PNAU joined online from various colleges, research stations, KVKs, and our PG and PhD students who are the stakeholders for this Endowment lecture series being organized by each department every week with the sponsorship from the Dean SPGS. So coming to the formal vote of thanks, let me first thank our university and our respected Vice Chancellor, Dr. V. Geeta Lakshmi, Madam. So we thank on behalf of the Department of Soil Science and Agriculture Chemistry, Directorate of Natural Resource Management, jointly with the Dean School of Postgraduate Studies, for permitting us to conduct this endowment lecture series on soil mapping with the sponsorship from the endowment. And we thank our registrar for about to join today in the absence of Madam and to deliver the presidential address. Again, due to an official commitments, pressing official commitments, he would not join. However, when we meet registrar along with our chief guest and director NR, we discussed and interacted with the speaker. Then coming to next our Dean SPGS and his office, coordinating with us for the conduct of this particular program. As soon as we submitted the proposal with the consent of the speaker, they have immediately got approval from the university and also sanctioned the grant for conducting this lecture in a successful manner. So we thank profusely the Dean SPGS and his office on behalf of the department and the directorate. And coming to the speaker, I'm very proud to say that he is our teacher. So you may wonder that Saras entered AIRS in 1976. How he was a teacher. Actually, he was a teacher for us, especially for ICAR fellowship coaching classes. Actually, during that time, Dr. Selvakumari Madam was the UG coordinator for us. And Madam used to get help from sir, especially to teach pedology, soil synthesis, and the soil survey. The same flow and the same tempo what I heard in 1984. Now I learned now. But actually, yeah, because why? Because many times we talk Newton management, all applied technology and all that. I think for the past 1.5 hours, actually, soil, soil, soil. That was the term it has been went on in each and every slide. And I was also just noticing the expressions of seniors and also my own students. And I also got some messages from out campuses also. So really, it was an excellent and a very good sensitization to all the students and faculty regarding soils. That's what I can say. So under his coaching SIP and with a team of teachers in my batch, that is in 85 UG batch, two of us got national level rank in ICAR Junior Fellowship. One is myself. I took national second, and another one is my classmate, Dr. Ratnasamy, who was retired in 21, the national sixth rank. So that was the scenario. So this is, and again for our batch, from our batch, the fellowship increased from 400 to 800 rupees per month. So it was spoken all along throughout that we got a double benefit from our batch. So that was the scenario. So really it was wonderful. And as soon as actually how I fixed sir as a speaker was, sir has called me on a Sunday and asking some clarification regarding the TN Soil Doc mobile app developed by the Department of Remote Sensing and GIS, mainly by Dr. Raghunath. So in that, sir had, he has gone through and he has got some clarification. So he called me and just asked. Then I contacted Raghunath and he spoke to him and he just clarified. So that time it struck me, definitely will utilize sir's speech in one of the lectures. It's so happened that Endowment lecture series has been announced and I submitted a proposal with sir's consent. So on this moment, at the age of 77, standing in the podium for about 1.5 hours, really we have to give an applause for his motivating lecture. And not only that, I want to expose, as he said, we should not restrict our knowledge with Tamil Nadu soils alone. We should have an overall scenario of the entire country. So he has brought from Himalaya to Kanyakumari. I think you will agree with me. So at this moment, I profusely thank you, sir, for having accepted our invitation and came over here in person and patiently delivered this lecture and for the benefit of all of us in the university. Thank you once again, sir, for your gesture and your acceptance. So I thank all the senior soil scientists. So as soon as sir has arrived, we have also informed all our soil scientists through email and through WhatsApp. Thank you all at this stage. You are all be with us. It is the kind of support to every one of us in the department. I profusely thank our former dean, Dr. K.K. Krishnamurthy sir, who was on time joined in the online. That's very interesting to notice. And Dr. Agustin sir, our teacher, and our Dr. Rakipan sir, who served a lot in our Sugarcane Bidding Institute at head of the division, and our former professor head, Dr. Muthuvel sir, who taught us pedology in PhD. So I profusely thank all the senior soil scientists and also other soil scientists who are joined online. And I thank the Professor Education, Dr. N.O. Gopal sir, and also Dr. Vanita, to whom we used to frequently contact for the arrangements, and they supported a lot, and also all the office staff of the Dean SPGS. And I thank all the faculty members of the department. So as usual, as a protocol in the department, we used to form many committees and served as conveners, and each and every one of us took their own effort in completing the task and we made it very successful. So I thank all the faculty members of the Department of Soil Science, the supporting staff for their nice arrangements. And at last, but not the least, the main audience, the PG and PhD scholars, especially of the Department of Soil Science and Agricultural Chemistry, and also from various other disciplines who are with us as because you are the stakeholders and your response is the success of the lecture. So I thank each and every one of them. So I thank all the supporting staff of the department also for their support in conducting the lecture. So once again, I thank one and all who are all associated with conducting this lecture in a very successful manner. Thank you very much.